And in part, that's because, if you think about it, we're bathing the brain in a chemical. This elaborate circuit made out of 1,000 different kinds of cell is being bathed in a substance. And that's also why perhaps most of the drugs, if not all on the market, can present some kind of serious side effect too. Now, some people have gotten some solace from electrical stimulators that are implanted in the brain. And for Parkinson's disease, cochlear implants, these have indeed been able to bring some kind of remedy to people with certain kinds of disorder. But electricity also will go in all directions, the path of least resistance, which is kind of where that phrase in part comes from. And it also will affect normal circuits as well as the abnormal ones that you want to fix. So again, we're sent back to the idea of ultra-precise control. Could we dial in information precisely where we want it to go? So when I started in neuroscience 11 years ago, I had trained as an electrical engineer and a physicist. And the first thing I thought about was, well, if these neurons are electrical devices, all we need to do is to find some way of driving those electrical changes at a distance. If we could turn on the electricity in one cell, but not its neighbors, that would give us the tool we need to activate and shut down these different cells to figure out what they do and how they contribute to the networks in which they're embedded. And also, it allows us to have the ultra-precise control we need in order to fix the circuit computations that have gone awry. Now, how are we going to do that? Well, there are many molecules that exist in nature which are able to convert light into electricity. You can think of them as little proteins that are like solar cells. If we can install these molecules in neurons somehow, then these neurons would become electrically drivable with light, and their neighbors, which don't have this molecule, would not. There's one other magic trick you need to make this all happen, and that's really to get light into the brain. And to do that, the brain doesn't feel pain. You can put, taking advantage of all the effort that's gone into the internet and telecommunications and so on, optical fibers connected to lasers that you can use to activate, in animal models, for example, in preclinical studies, these neurons and to see what they do. So how do we do this? Well, around 2004, in collaboration with Georg Nagel and Carl Deisseroth, this vision came to fruition. There's a certain alga that swims in the wild, and it needs to navigate towards light in order to photosynthesize optimally. And it senses light with a little eye spot, which works not unlike how our eye works. In its membrane, or its boundary, it contains little proteins that indeed can convert light into electricity. So these molecules are called chanorodopsins. And each of these proteins acts just like that solar cell that I told you about. When blue light hits it, it opens up a little hole and allows charged particles to enter the eye spot. And that allows this eye spot to have an electrical signal just like a solar cell charging up a battery. So what we need to do is to take these molecules and somehow install them in neurons. And because it's a protein, it's encoded for in the DNA of this organism. So all we got to do is take that DNA, put it into a gene therapy vector, like a virus, and put it into neurons. So it turned out that this was a very productive time in gene therapy, and lots of viruses were coming along. So this turned out to be fairly simple to do. And early in the morning, one day in the summer of 2004, we gave it a try, and it worked on the first try. You take this DNA and you put it into the neuron. The neuron uses its natural protein-making machinery to fabricate these little light-sensitive proteins and install them all over the cell, like putting solar panels on a roof. And the next thing you know, you have a neuron which can be activated with light. So this is very powerful. One of the tricks you have to do is to